Welcome to our first podcast on the blood. What we're going to cover in this unit is going to be erythrocytes and granulocytes. So the erythrocytes, we're going to look at the molecular components, not just the hemoglobin inside, that's the oxygen binding molecule, but also the cytoplasmic and the plasma membrane components that help give our erythrocytes their characteristic shape. We're also going to look at the cellular characteristics and the histological characteristics that will help us distinguish our three types of granular leukocytes: the neutrophils, eosinophils, and the basophils. So our blood is characteristically going to be about a pH 7.4. Remember, that's physiological neutral, and the volume for an average adult is going to be around 5 liters of total volume, which accounts for approximately about 7% of your total body weight. Now, when we look at a unit of blood, we're going to see that we have the formed elements, which consist of our erythrocytes, leukocytes, and platelets, and then we have the plasma. So typically, we're going to be seeing that we've got approximately 55% of that volume of blood is going to be plasma. Then we're going to have 45% that are the other elements. So we're going to have 44% or so. The majority of this is going to be red blood cells. So you could really count red blood cells as 45%. White blood cells and platelets, each are going to be less than 1%. I know when you add that up, you get 101%. But largely, we have got a minor contribution to the hematocrit that's contributed by the white blood cells and the platelets. So you can either do a hematocrit using a plasma tube, or you can use a capillary tube. Regardless, as you separate these out with a centrifugal force, the most dense elements, red blood cells, sediment to the bottom. Plasma, the least dense, is going to be buoyant and rise to the top. And situated between, you have the fraction of white blood cells, leukocytes, and platelets, which is referred to as the buffy coat. And I think you can see here that compared to the total volume, the buffy coat layer is only a fraction of a percentage. Now, when we think of blood, we certainly realize that we're transporting oxygen to our tissues, but we're also going to transport carbon dioxide back to the lungs. Now, the carbon dioxide is going to be transported by and large by the bicarbonate ion. We talked about that in phys, and we're not going to go in depth about that carbon dioxide transport as well, but it's transported as the bicarbonate ion. We're also going to transport nutrients, waste products in either direction. But remember, we also are going to regulate our body temperature. We can either dilate vessels to the skin and radiate more heat, or we can constrict those vessels through our arteriovenous anastomoses, keep the blood from getting out to those extremities, and therefore retain a lot of our core body heat. Because of the proteins in the blood, we're going to maintain this osmotic balance of tissues with our oncotic pressure, oncotic referring to the protein component. This is going to be a major migratory pathway for leukocytes as they circulate throughout the body. And then, as we also learned in physiology, we've got the coagulation factors present in the blood to help with the process of hemostasis in times that we have a vascular injury. So the plasma, just as a review, 99% of plasma is water. We also have the protein part. And again, this is going to be a fraction of that total volume, and that should be 1%, not 9%. But that protein portion, as well as the salts, ions, nitrogenous compounds, this is going to be a very small part of what we see for the total composition of the plasma. So the proteins that we see, we've talked about these again in physiology. We have the albumin fraction, which is going to be a carrier molecule. It also helps to maintain this colloid or oncotic pressure in our blood vessels at tonicity. We're going to have the globulins, the alpha and betas, but principally the gamma globulins or the antibodies. We have complement proteins, fibrin. We have, fibrinogen, the inactive form of fibrin. We have prothrombin. All of the other clotting factors, especially those involved in the intrinsic pathway, are present. And then we also have complement proteins that are in their inactive form that, when activated due to an immune response, can lead to the destruction of microorganisms. In addition to proteins, we have the lipoproteins. We can have the chylomicrons, that transport that we see from our intestines, transporting that back to the liver. We have the LDL, we have the VLDL, and then we have the HDL as well, the high density lipoproteins. Remember, high density is the good lipoprotein, and the LDL is the one that deposits cholesterol from the liver to the cells. And so the LDL is the bad form of cholesterol. This is just an illustration showing what's going on with the complement cascade. We have all these different complement components. We can see in this particular illustration the attack complex that is formed in the end that inserts itself into the membrane of a pathogen and releases material, therefore lysing the particular pathogen. We also have antibodies when they're opsonized on the surface. We can have various components of the complement pathway that are going to enhance opsonization. And then we have other components that are going to go off that help in the inflammatory response of tissues. Here we have an illustration of our lipoproteins, the LDLs, the HDLs, the VLDLs. All of these different lipoproteins are important in the trafficking of water-insoluble fats, fatty acids, and cholesterol. So now with erythrocytes, typically when we look at our red blood cells, they are going to have the biconcave shape. This is going to maximize the surface area to the volume of the cell. This ensures that from the surface or plasma membrane to the deepest molecule of hemoglobin is going to be equidistant anywhere you go in the cell. If you had a round cell, there would be hemoglobin molecules in the very center of that cell that would not be able to have oxygen diffuse on and off. The average diameter is said to be around 7.5. I like the 8 micron term. It's a nice round number that we can hang on to. And the average lifespan of a red blood cell that is going to be anuclear is 120 days. And in that 120-day time, that's sufficient time for a single red blood cell to make over 100,000 trips through the entire body. Some demographics: males typically have a higher hematocrit than females, and again, that's because of the higher, typically the higher muscle mass that's going to require oxygen for males than females. And also individuals living at higher altitudes where you have a lower oxygen content, the erythropoietin is going to be expressed. It will increase your hematocrit so you can be more efficiently transporting the little oxygen that's present. As we said, your mature erythrocytes are going to be anuclear. They have no organelles to speak of. However, they do have various enzymes and proteins associated with the membrane and the cytoplasmic phase. Carbonic anhydrase is the enzyme that's going to convert carbon dioxide into carbonic acid. This then dissociates to form the bicarbonate ion, and we'll be able to transport and process that as we transport that to and from the tissues to the lungs. Now, when we look at the proteins that are present, there is a band 3 protein, and this is going to be a channel that transports the bicarbonate ion across the red blood cell membrane as it shifts the chloride ion in. So this is going to be our chloride shift. And when we get to the lungs, remember, we do the reverse chloride shift. Chloride is able to come in, and the bicarb comes in and the chloride goes out. That's the reverse that we hit when we get to the lungs. Our typical picture on the left, we have our scanning electron micrographic picture showing the 3-dimensional surface, the biconcave disc. And on the right, we have our typical light micrograph of peripheral blood showing somewhat of an empty donut shape to our red blood cells. It's less dense in the middle than it is on the rounded edges. We can also see a platelet in that peripheral smear. Now, we mentioned the red blood cell and hemoglobin. Hemoglobin is the molecule that's going to transport oxygen. And for the hemoglobin molecule is comprised of 4 protein chains. Each one has an iron-binding heme group in the center. So for each hemoglobin molecule, you can bind 4 molecules of oxygen to the iron that is bound. So when we look in the lungs, the movement and loading of oxygen to hemoglobin is driven by diffusion. No energy is going to be used. So in the lungs, oxygen is going to be loaded on hemoglobin. And when we get to our tissues, oxygen is going to be unloaded. And this is really a balance that we have with carbon dioxide. And remember, that's the pH that's really affected here. Carbon dioxide is affecting the pH, which assists in either the loading or the unloading of oxygen. So in areas where we have low oxygen and higher CO2, that's going to favor the unloading reaction, and that's going to happen down in our tissues. Now we also see that nitric oxide can bind to hemoglobin. Nitric oxide, we know, is a potent vasodilator. When we have individuals that are suffering from blockages of coronary arteries, they're beginning to have the angina, the heart attack symptoms, they can put a nitroglycerin tablet under their tongue. That is quickly converted into nitric oxide that systemically goes through their body, but certainly it dilates the vessels in the heart and gives them some relief from those symptoms. Probably one of the most tightly bound molecules to hemoglobin is going to be carbon monoxide. And as a competitor for oxygen, once carbon monoxide binds, it's going to be very difficult to get that off. You're going to have to give a patient pure oxygen so that as the stoichiometry, the on-off of carbon monoxide happens, you overwhelm the system with oxygen so that the chances of carbon monoxide binding more tightly are offset by the vast numbers of oxygen that are competing for that site with a lower affinity. And that's the only way you're going to help someone that's suffering from carbon monoxide poisoning. Here we have an illustration of our hemoglobin molecule. You can see in this one we have our 2 beta chains, 2 alpha chains, heme group in the middle where the iron is bound, and where we have the oxygen. In this case, we are looking on the right at sort of a space filling model, and we're looking inside at the heme group, and we can see that hemoglobin actually changes conformation based on whether an oxygen is bound or an oxygen is not there. So you can see the shift from oxyhemoglobin to deoxyhemoglobin when we do not have an oxygen. And you can actually see in this illustration and animation the conformational change. Now remember, we do have these different chains that are present to make up hemoglobin. 96% of the adult population will have the alpha-2 beta-2, but we also have a small percentage that are going to have the alpha-2 gamma-2 that have the alpha-2 beta-2 chains of hemoglobin. However, 2% of the population are going to lack the beta chain. They're going to have the delta chain. Now, when we are all fetuses, we will have fetal hemoglobin, so the 2 alphas and the 2 gamma chains. These are going to create a much higher affinity for oxygen than our alpha-2 beta-2s or the other adult forms, and therefore the oxygen shift, the loading curve that we have is going to be shifted toward the left. And remember that from phys, it's going to be in favor of that fetal hemoglobin, and it's going to assist in moving oxygen across the placental membrane from maternal blood to fetal blood. And in fact, in the adult population, we do have 2% of the population that still, as adults, express the 2 alpha and 2 gamma chain form of fetal hemoglobin, even as adults. In addition to hemoglobin, there are many other molecules, proteins, carbohydrates that contribute to the shape and the structure of red blood cells. And as we can see from this cutaway, there are a number that are transmembrane proteins that have extracellular domains, such as the glycophorin A and C. We also have the band 3 protein that we've already mentioned as an ion transporter. But then on the cytoplasmic face, we have a lot of structural elements that help maintain the biconcave shape of the red blood cell. Now, band 3, in addition to being the ion channel, also has a cytoplasmic domain that is able to interact with ankyrin. And ankyrin is a protein that is going to cross-link band 3 to this underlying hexagonal network that is going to be comprised of spectrin, and spectrin also is going to be able to interact with the actin cytoskeleton and therefore create this underlying network that's going to create and pull the plasma membrane down and pull it into the shape of the biconcave disc. So here we can see spectrin. It is a twisted dimeric molecule. We can see actin is involved. We also have band 4.1 that's going to help to cross-link all these molecules together, and all of these interact with band 3, glycophorins, to hold them in place in the membrane, and then, as I mentioned, pull the membrane downward into that biconcave shape. Now, we also have proteins that do not play a role in the shape, but they do function as part of blood groups. And we know that we have the different antigens. We have the H antigen, which is represented as the O-type phenotype of blood, the A and the B antigens, and then we also have the blood group of the Rh factors. We have the 3 antigens C, D, and E. 80% of Americans are Rh positive, and the most common blood type among Americans is A positive blood. Now we turn our attention to the white blood cells, or the leukocytes. There will be many fewer leukocytes in a volume of blood than there were red blood cells, in the order of maybe 5,000 to 10,000 white blood cells per unit of blood. And really, for the white blood cells, the circulatory system is only a highway system with which leukocytes move from place to place. Now, leukocytes, unlike red blood cells, can leave the circulatory system. This process is called diapedesis. White blood cells actually squeeze between endothelial cells, force their way past the basement membrane, and into the interstitial spaces. Diapedesis is not unidirectional. This can also occur as white blood cells make their way back into the circulation. So our leukocytes are characterized into 2 broad groups. First, we have the granulocytes, and secondly, we have the agranulocytes. This is based on the fact that when light microscopists looked at the staining pattern of these cells, they saw a granular appearance. This was conferred by specific stains and looking for specific granules, and some quite large. So based on this staining pattern, we have the neutrophils, which have a neutral staining pattern, eosinophils that are somewhat reddish, and basophils that are dark blue. Now, the 2 types of leukocytes that lack these specific granules, now they will have smaller granules that are called azurophilic, and it's based on that particular stain. These are in fact lysosomes, because all of these cells, granted the lymphocytes to a much lesser degree, but the granulocytes and the monocytes, these are going to contain lysosomes. And so to say they're completely lacking of granules is a little bit of a misnomer. The agranulocytes lack these larger specific granules that we detect with the stain, and hence give them the name. Our agranulocytes are the lymphocytes, the B's and T's, and the monocytes that we call macrophages when they leave the circulation. In this image, we can see our lymphocyte, mostly nucleus, very thin rim of cytoplasm. The indented nucleus of the largest of our peripheral blood cells that we're going to see, this is the monocyte. Then as we look at our granulocytes, we're going to see that we can have different shapes and patterns and sizes to the nucleus, different staining. These are the smallest granules, somewhat of a neutral staining color. We can see 3 lobes to this nucleus. This is a neutrophil, or a polymorphonuclear leukocyte. Here we can see a much more granular appearance, bilobe nucleus, reddish in color. This is our eosinophil. And then lastly, we have such large blue granules in this cell, it's almost difficult to see the nucleus. And this is indicative of a basophil. So let's first look at our granulocytes, and specifically the neutrophil. These are going to be the most abundant of all of our white blood cells, and because of that abundance, these are going to be the first ones to appear at the site of a bacterial infection. These are our short hitters, remember, and the average lifespan of a neutrophil is on the order of 12 hours. They're first to arrive, and they're the first ones to die. Again, we have these lobes to our nucleus, and the older the neutrophil, the more lobes they can have, upwards to 5 to 6 lobes. And we can see they're somewhat larger than our red blood cell. They're somewhat larger than our red blood cell, approximately 8 microns. These are on the order of 10 to 12 microns will be the average range. So slightly larger than we see with our red blood cells. Now, the other indicative feature that we see of the neutrophils is the fact that in females, the condensed X chromosome, the condensed second X chromosome, the bar body, this is going to be visible almost as a drumstick-like region on the neutrophil, the nuclear material of our neutrophils in females. And so as we look at 2 of our neutrophils, we can see the lobes to our nucleus, possibly 4 lobes here. Here we have 1, 2, 3 lobes. And you almost have this little drumstick-like projection There's that one there, and I'm not sure if it's that one or this large one. I'm sort of inclined to go with this smaller one. That's going to be the bar body, and that's going to be the condensed X chromosome of the neutrophil. Now, we mentioned the specific granules. Certainly the neutrophils have these, and many of these are going to be antimicrobial, and we'll talk about some of these. For instance, we may have lysozyme. This is going to be an antibacterial agent. Certainly with permeability increasing protein, they call that BPI. We have tertiary granules, gelatinases, cathepsins, and these are going to be proteases that assist in degrading the basement membrane as these cells are diapedesing in and out of our circulation. Now, as we look at diapedesis, the way that white blood cells in general, and we're just looking at neutrophils in this particular instance, the way they're able to leave the circulation, remember, they're flowing very rapidly through the bloodstream. And so it's not as if they can just reach out, grab a hold, and diapedese their way through very easily. There's going to be steps to this process. In the first step, neutrophils are going to have receptors. And these receptors are going to bind to selectin molecules on the endothelial cells. And this binding is going to be this first slowing down of the neutrophil, and it's called rolling. So as the selectin molecules are bound by neutrophil receptors, they begin to roll along the endothelial surface. The next step, we then have our endothelial cells express ICAM1. ICAM1, intracellular adhesion molecule 1, will be able to bind with integrins on the neutrophil cell surface. And this binding has a much greater affinity. It's going to bind with more strength. And so this is going to cause the neutrophil to stop. Whenever you have this interaction taking place rather than the selectin binding, now you've had rolling, which is slowing down the neutrophil, and the integrin ICAM interaction is going to stop the neutrophil. And at that point, now the neutrophil can diapedese through in between the endothelial cells. So here we have our selectin molecules on the cell surface. This is E-selectin for endothelial selectin. Here we have our selectin receptor. And really it's going to be the carbohydrates interacting with the selectins that are going to slow the neutrophil as it's rolling along. Now we have the ICAM. This is expressed on the endothelial cell, binding to our integrin. There's our alpha and beta subunits. It's alpha L, beta 2, and this is called LFA1, this particular integrin that's going to stop the movement of the neutrophil. Blood is still flowing through the vessel, but now we've stopped the neutrophil. Diapedesis occurs, and we can release our enzymes in the basement membrane interstitial spaces, so now the cell can easily migrate in the direction of the pathogen. Now, a few of other things that happen with our white blood cells as we look at our granulocytes and our macrophage monocyte population. These are going to be phagocytic cells. They're going to internalize pathogens through phagocytosis. They will form a membrane-bound vesicle with the material inside that's called a phagosome. Then we have other vesicular traffic in the cytoplasm, this lysosomes. These have degradative enzymes. When those fuse with the phagosomes, you create a phagolysosome. Now the enzymes are released inside with the pathogen, and that's going to cause the destruction of whatever material is inside the phagolysosome. Now, in some cases, these white blood cells are able to produce what is called a respiratory burst. And so we are going to form hydrogen peroxide, hypochlorous acid, and when these are brought together, these are going to form a superoxide ion. So these materials are going to be injected into that phagolysosome, and this is going to assist in the destruction of the microorganism. Now, all of our white blood cells can produce and secrete chemokines, and these are going to be chemical agents that can signal for increased migration of leukocytes. It can enhance the dilated state of a blood vessel, many cellular responses, and one of the chemokines that are produced are the leukotrienes. And the leukotrienes are going to be secreted and lead to basically an inflammatory response. Part of that response is going to cause endothelial cells to be leaky, but that's also going to be the recruitment of other immune cells to the area of the infection. Now, because of the phagocytic activity of our granulocytes and macrophages, once you fill these cells with enough pathogenic material, once you basically have filled the cell with phagolysosomes and destroyed those pathogens, now the neutrophils die, the macrophages die, and you're left with material that we simply call pus. So in this illustration, we can see an example of how this process happens. Here we have a phagocytic cell. It has different receptors. The C3b, remember, this is a complement protein that's bound to antibodies that have opsonized this particular bacterium. Here we have a C3b receptor on the surface of our phagocytic cell. Here we have an Fc receptor, much like what you might find on a mast cell that has the receptor for the Fc fragment of IgE type antibodies. And once you bind these materials, then endocytosis or phagocytosis occurs. And here we can see the pathogen brought inside the cell. And here we're in the process of forming our hydrogen peroxide as these vesicles are dumped in with this material. And now we have destroyed the pathogen. We are going to begin to process this. In many instances, we're just going to leave this waste material inside, and it will actually cause the cell to die if there's enough of it in there. Otherwise, the cell can resorb some of this material, exocytose the debris to remove that from the area. Now, the next granulocyte are the eosinophils. And again, these are going to have about the same diameter that we see with the neutrophils. However, their granules are going to be dark red to pinkish in color. Many fewer of these in the white blood cell population than we saw with the neutrophils. These are going to survive less than 2 weeks, so they're a little bit longer lived than we saw with our neutrophils. Here we see an illustration with a bilobe nucleus, the pink granules, and then we can see our electron microscopic stain. It looks like we have 2 nuclei, but remember this is a plane of section showing the 2 lobes that appear to be disconnected. The arrows are pointing to some of the specific granules, and they have a very defined structure that we're going to look at in just a minute. Here we have an immunohistochemical stain, or a histochemical stain, if you will, of peripheral blood. You can see our red blood cells quite nicely. And then the very large pink granules, bilobe nucleus. Cell is just a bit larger than our red blood cells, 10 to 15 microns versus 8. And so this is an eosinophil. Now again, as we look at these specific granules in the granulocytes, we're going to see that they're really comprised of 2 portions. We have the internum and the externum. These are going to be oblong structures, about 1.5 nanometers in length. That shouldn't be millimeters, nanometers in length. And the internal crystal-like core is called the internum. And these are going to be proteins that are essentially stored in these granules. We have major basic protein, eosinophilic cationic protein, eosinophilic derived neurotoxin. All of these are going to be released again in response to pathogens. And then the externum is going to be that portion that surrounds that internal crystal-like structure. It's less dense material, so it's not going to be as dark as we can see. And in many instances, the internum is going to have degradative enzymes. Those are definitely going to be present. And then other vesicles that we have that are not going to be these specific granules, much smaller, these are going to be our lysosomes. So here we have an illustration of transmission electron micrograph. We can see this oblong structure. We can see the crystalline core of the internum in the middle. We have our externum on the outside, and you can see many of these present in these eosinophils. Now, these are going to be phagolysosomes that we have. We can see the material and the debris within them. Lysosomes may be as small as this that you see here. Now, eosinophils are primarily going to be involved in the destruction of parasites, but they will, as with many of the other granulocytes and macrophages, they are going to assist in eliminating antigen-antibody complexes. So as pathogens become opsonized with antibodies, locked into these big rafts and mats, these are going to be eliminated by eosinophils. So eosinophils, like other immune system cells, are going to be chemotactic. They can follow these chemical trails, and molecules such as histamines, leukotrienes that we mentioned earlier, eosinophilic chemotactic factor, which is produced by macrophages, these are going to attract the eosinophils to the area. And as we mount an immune response, and as we get to a concentration level where where it's very high, we know we have arrived at the scene where we encounter the pathogens, then degranulation occurs. And this is somewhat of a generic term, which means a very rapid, robust release of the materials from all of our vesicles and granules at one time. This term came about from light level microscopists that saw granular eosinophils, and then after they were immunogenically challenged, the granules were gone, so they degranulated. And so in this case, one instance, we have major basic protein, eosinophilic chemotactic factor, that's the ECP. These are released into the pellicles, this sort of cuticle that surrounds the worms and parasites. It's a very tough collagenous cuticle typically, and these factors help to form pores through the pellicles, and this allows the other substances that are produced, the superoxides, hydrogen peroxides, to pass through that hard protective coating, the pellicles, and to get inside and destroy the parasite. Now, eosinophils are also going to be involved in sort of a negative feedback means, because as mast cells, for instance, secrete the histamines, leukotrienes, heparins, eosinophils are going to function in inactivating many of these inflammatory molecules. And so they downregulate inflammatory responses, and that way we sort of have the negative feedback to keep the inflammatory response in check. Lastly, again, eosinophils are going to be phagocytic, those antigen antibody complexes, and so we can find a lot of this material is a phagolysosome. The waste material that is still stored inside yet to be eliminated, these are called endosomes. Basophils, our third type of granulocyte. These are going to, again, be about the same size. Typically, we can have the bilobe nucleus, but we're not really going to see the nucleus and the stain of the nucleus, because these are going to have the largest of the specific granules and extremely dense and dark in color, either blue to black. These are going to be the fewest of our granulocytes as far as percent that we will see, but they sometimes have the greatest effect on human body and function as they relate to extreme allergic reactions, those anaphylactic type responses. Now, of the three, these are the longest lived. They may live 1 to 2 years, so there's a very extensive range with which we can see lifespans of granulocytes. So this is our illustration. We can see a red blood cell illustrated at the lower left, 7 to 8 microns in diameter. And then again, the nucleus is somewhat obstructed because of the large blue granules, and that's even more the case as we look at our peripheral blood stain. We see our red blood cells, larger granulocyte, and you are hard-pressed. I know I can't. Hard-pressed to see even where the nucleus might be. You might see it looping around like this. But when you have a total obstruction of the nucleus, dark blue to purple granules, you know you're looking at a basophil. Now, the specific granules of the basophils, as we talked about in physiology, we're going to have the heparins, the histamines, the chemotactic factors for the eosinophils and the neutrophils, really assisting in mounting that immune response. They will also have lysosomes as they phagocytose material, much like we saw with the neutrophils and eosinophils. Now, the mast cells, these are what we call basophils as they leave the circulation. So these mast cells now are going to initiate largely our inflammatory responses, and this happens often because of IgE antibodies. These IgE antibodies can be produced in response to certain allergens, and we refer to them as allergens because they are antigens that cause an allergic type of immune reaction. So these IgE antibodies, they're going to be inappropriately bound to these IgE receptors on the surfaces of mast cells. They can be basophils too, but really we're talking about the mast cells outside of the circulation. And when you have a secondary exposure to this particular allergen that is recognized by these antibodies, that's going to cross-link the antibodies, bring them close together, because remember, our antibodies are bivalent. And as you bring the antibodies closer together, that's going to bring these IgE receptors closer together on the cell surface. That's going to lead to signaling that cell that that pathogen is present. It's going to cause rapid degranulation of that cell. Leukotrienes, they're going to be released, the heparins, the histamines. And remember, again, from physiology, if this happens on a systemic level, and if there's enough of this released, enough mast cells, basophils to degranulate, that can plummet your blood pressure. You can head for an anaphylactic response, and if not treated, death may ensue.